We left off in the middle of chemistry. Today we're going to be talking about chemistry and then moving on to thermodynamics. And I kind of ask people for feedback, and sometimes people will send me emails. And I try and listen to feedback, so let me know. And I keep getting this over and over, so I'll do it. And it's to sort of go over what we're going over. So we're going to start with chemistry. And then we're going to go over the four sort of functional building blocks. And you all remember what they were? I'll just start.
Fats, lipids, proteins.
All right, good. I heard lipids,
Carbohydrates.
fats. What else do we have? I'm just going to keep writing, and you can read it aloud. Carbohydrates and nucleotides. All right. And then we're going to move into thermodynamics. So this is kind of the second half, and I'm really just going to be able to get through an introduction on thermodynamics. We're going to talk a lot about proteins today, and then more later. But we're just going to do sort of an intro on chaos, and that sort of thing. So that's roughly what we're going over, and I will try and point out what I think is more important as we go on. All right, so this is, I think, where we ended last time. So I know that some of you were here, some of you weren't. But who can tell me what the structure is? All right, so we're going to break it down, and the whole point of this is using our knowledge to answer questions without memorizing them, conceptually answering things. And so I'm going to keep trying to get back to that. What's this group here?
Amino.
Amino, okay. What's this group?
Carboxyl.
It's a carboxyl, but what's the full name for it? This is a carboxyl group, you're right. What's the full name for it?
Carboxylic acid.
Carboxylic acid. Okay, so we have an amino carboxylic acid. So it's N.
Protein.
So, okay, not
It's
quite,
amino acid.
right? But you're getting, it's an
Amino
amino
acid.
acid. This is an amino acid. This is the basic structure of amino acid. Okay, and then the R group is what?
Side chain.
The side chain. Okay, good. So here are the different side chains, all right? So what this picture is doing is it is lumping the side chains into different broad categories, right? Nonpolar, polar, electrically charged. These are false distinctions, right? It's a spectrum of things. God is not lumping these into categories, we are. The way that they're acting is just the way that they're acting. But it's useful to think about them in certain categories because it lets us kind of predict what they're going to do. So the first thing I want you to notice is that this structure here is not the same as this, right? There's 3 hydrogens here, and this carboxyl group has lost its hydrogen. So it's getting at the first point of environment matters, okay? In a physiologic environment, in physiologic pHs, this is what you'll see. So we're already getting at this nature versus nurture thing where environment matters every step of the way. Okay, so that being said, look at the different groups and kind of point some things out to me, right? Some of you know what makes things polar or not or whatever, but just sort of, even if you don't know that, just kind of tell me what are the main differences that you see in the side chains between the polar and the non-polar.
Some are aromatic and some are not.
So lots of aromatics up here, a couple aromatics here, and so, okay, good, lots of hydrocarbons, right? Lots of hydrocarbons here. What is sort of in here that's not so much in here?
Extrapolar groups like amino or
The OH.
OH.
Yeah, it's polar groups, like amino groups, right? And SH here. So now a couple of things. If we had to understand that these are not rough distinctions, in this polar group, what do you think is the least polar?
Serine.
Why serine? That's a fair guess, but
This is the smallest.
it's the smallest. Okay, so it actually has a lot of relative charge for its small size. It's probably actually more polar than 3amine, but I'm just sort of making rough assumptions there. It would be cysteine. Okay, so if we had to come back and look here, what's the most polar in the non-polar group? It could be a couple of them, but I'm kind of getting you all to think so that you don't need these distinctions. You can just look at molecules and guess what's going to happen. Where is the relative charge differences up here? So these are all carbons, right? Nothing but hydrocarbons, all right? So it's not going to be here. Hydrocarbons. A nitrogen, right? Nitrogen has more of an affinity than carbon does, so we have a relative polarity here. Sulfur, again, when we see these groups, we know these two are going to be the most polar in this nonpolar group. I just want to get you guys thinking, okay, what else stands out in this one? Which amino acid here is different than every other amino acid? One of them is different than all the other 20 in one bond that it can make.
Proline.
Proline, right? What is proline doing?
It's binding back to the amino
That's
group,
right.
the R chain.
It's a secondary amino. So do you think proline is going to look the same as the rest of them when you create a protein? No, right? So we can look at this and we can make predictions about what's going to happen, and you'll be right. You'll be right a lot. When proline forms, it forms a kink. It's rigid, right? These things can whip around. This is more rigid. It can't move. It's going to force a conformation as we're throwing those amino acids into a growing protein chain. So there is value in understanding chemistry. It's kind of the idea here. And the other is that this is a relative scale, right? We can draw these lines. They don't really exist. So when we're creating these proteins, there's a lot of flavors in the way that they can sort of act. All right. So these are amino acids. And as you can see, they're just whipping around in the cell, right? They're flipping around in the cell. And they look like this. They're actually fairly reactive, right? They have these relative charges. And this is a lot of charge for a small molecule. So it is active. When it is being formed into a protein, what is happening? And in a protein, how many positive and negative charges, although discounting in the backbone, how many positive and negative charges are in a protein chain? Whether there's one amino acid or there's 1,000 amino acids, how many are there?
Should be net neutral, right?
Net neutral, yes, that's true, but net neutral doesn't so much matter because even though it's net neutral, this is very active when it's flipping around, and so is this. So even though it's net neutral, you have a lot of potential reactivity. It really matters more the number of charges than the net charge when it comes to reactivity. So in a long in one amino acid, how many charges are there, not counting the side chain?
2.
2. In 1,000 long amino acid protein, how many are there? 2, right? Because that's what we're going to get into next, that when this one bonds to the next one, it's going to come here and these two charges are going to sort of cancel out. So flipping around, these are actually very active, and these are used a lot to make things, right? What does tryptophan make? That's important to us as psychiatrists. Serotonin, right? And so we talked a little bit about how we make things, right? We take things in and we turn them into other things. What is it that turns tryptophan into serotonin? What are the things, not specifics, just generally, what turns things into other things generally?
Enzymes.
Enzymes, which are
Proteins.
proteins, right? They're made of these things, which come from
Amino.
amino acids. And where are the instructions?
The
DNA.
In the DNA, right? Okay, we're getting back to these sort of same concepts over and over and over. All right, so if we have, now you can imagine a long amino acid, there's relative things that these things want to do. The polar side chains are going to want to flip out to the outside, the nonpolar are going to want to be on the inside, but you might have a polar and a nonpolar next to each other, and who knows how they're going to twist and tug and ultimately sort of form their final shape. And the other thing that I want to point out is that environment matters every step of the way, right? Relative charge in the environment matters, which is pH pretty much. Temperature matters, and we know that. With temperature, energy in bonds change, conformations can change, you can denature proteins. Lipid environment changes, so you put something in something that's more polar or more nonpolar, and the relative charges and attractions change. So just getting at this idea, it's complicated, it's complex systems, but it's still useful to make predictions based on what we know about basic chemistry. And what's the two things that I say mean everything when it comes to chemistry and biology in life? What matters, right, when it comes to physics? The physics that we care about, two things.
Charge.
Charge and shape. All right, shape and charge dictate everything. That's electrodynamics, that's all that we do. All right, so getting here, a few things, right? Look up there. What is this? What's L-tryptophan? What's this group? What's that? A carboxyl. It is an
Amino acid.
amino acid. Okay, you don't need to memorize them all. You can look at them and see what they are, right? And this is its side chain. All right, the first step in conversion. What is this? Tryptophan hydroxylase. What is it?
Enzyme.
It's an enzyme. Okay, good. It is called tryptophan hydroxylase.
So it's adding.
Thank you. Okay, that's what I'm getting at. Words have meaning, right? We throw these words out there. You can just memorize them, or you could actually take a second and be like, okay, it's tryptophan hydroxylase. I can see it in my head. It's an enzyme. That means that it's a protein. That means that it's amino acids, right? So we can start to think about these in real ways. They're not just words. It's going to add a hydroxyl group. Okay, where is that hydroxyl group?
On top of the chain.
In the 5 position, right? This is 1, 2, 3, 4, 5, right? So we've now made 5-hydroxytryptophan. Tryptophan hydroxylase. All right, the next step, which you don't usually need to know, and I'm not going to go into a lot of details here, is 5-HT decarboxylase. What do you think it does?
Removes the carbon.
It removes the carboxyl, right? Yoink! And we've got serotonin, right? Or 5-hydroxytryptamine. And then this is just the conversion, monoamine oxidase into 5-HIAA. All right, so I'm just trying to get you all relative size differences. Roughly how big is serotonin? Again, I wish I had more continuity so that we could kind of keep this going. But let me say that glucose is about a nanometer. A carbon atom is about an angstrom, or 0.1 nanometers. Or let's just say we're saying relative to an amino acid. How close is serotonin to an amino acid?
Pretty close.
It's pretty close. So it's roughly 1 amino acid, okay? This here is orexin, okay? It's a neuropeptide, right? Orexis means appetite. It is like, I wrote it down, 33 amino acids, okay? This is a, anybody remember?
Receptor transporter.
Not a transporter.
Oh, the receptor.
Receptor, okay, you want to guess how many amino
Receptor.
acids this is? Just a guess, I mean, it doesn't.
50.
So this was 33.
Oh, it's like a 700. No, 700.
I don't know if it's 7, 1, 2, 3, 4, 5, 7, yeah, it's 7.
300.
So like.
Yeah, right, 600, it's 600, right?
Yeah.
So the idea is, these are rough distinctions, but we can begin to understand relative sizes of what's going on. So the enzymes here that are working on this are massive compared to the shapes. And they're probably more 200, 300, it depends on the individual enzyme. This is a peptide, just by definition. Less than 50 amino acids, peptide, more than 50 amino acids, a protein, okay? That's really the whole point of all this. But also getting at this idea of how do we make things, right? We're eating an amino acid, and we're making serotonin, and we can determine what cells make serotonin by regulating the genes that they make. You don't make tryptophan hydroxylase, you don't make serotonin, okay? Every cell has this gene, but only the genes that express it are going to make serotonin. So that's how we decide what cell makes what. That's how our body makes things. It takes stuff in and decides how to make it through proteomics, through gene expression. All right, try and go fast here. All right, this is called a, okay, it's a special type of, does anyone know what this compound is? C double bond O? C.
Ester?
It's not an ester. If this was an O, it would be an ester.
Oh.
That's right. It's an amide bond, right? So this is good. I'm impressed by that. This is a special kind of amide bond. It's a peptide bond. And it's just a bond between 2 amino acids. So that's pretty much it. So a couple quick questions. I skipped this slide on electrophiles and nucleophiles, okay? In retrospect, I'm glad I did, because now I can get you guys to teach me what those things are. Okay, you have all the knowledge you need to understand how these things work. All right, so let's start with a nucleophile. What's a nucleophile?
Nucleuses.
Yes, okay, that's exactly the answer I wanted. We have the word nucleophile. Loves nucleus. Phile means love, nucleo means nucleus. Okay, what's the charge on a nucleus?
Positive.
It's positive. Why?
Because the electrons are sort of go externally. I don't know.
You're thinking, all right, so
Yeah.
it's positive. Why? The electrons are on the outside.
Mm-hmm.
What's inside the nucleus?
The protons.
And the protons have a,
Positive charge.
okay, so the positive charge is on the inside.
Uh-huh.
So that's why the nucleus is positively charged. So a nucleophile loves a nucleus which is positively charged. What's the charge on the nucleophile then?
Negative.
Negative, right? Because negatives attract. That's just, electrical sort of physical laws. So what's the nucleophile here?
The C-terminus. Or the
Okay, let's not
oxygen.
call it the C-terminus yet, but yes, that would be the initial, but we don't have a terminus quite yet. The oxygen. The oxygen that's negatively charged, right? So we know that electrons attack, and I like looking at words a lot, and I just want to sort of make an aside here that science has been male-dominated for way too long, like a lot of fields, and I would love for electrons to cuddle, and I think that's just a plug for we need more diversity in lots of things, but particularly science. So anyways, And this is complicated, it's going to flip over here, they're going to flip back, whatever. Ultimately, you form this amide bond. And, we talk about the N terminus and the C terminus. And, again, you can memorize these things or you can just understand the concepts. What are we talking about? How many N terminuses are there going to be? How many C terminuses are there going to be? One, yes, and that's where the charges are, right? Because as the next one comes in, yoink, this is no longer the C terminus, we've got a new one. In the ribosomes, we're just sort of throwing them in and we're extending them, whatever. All right, so another aside. Yeah, I talk a lot about words, and Paola, I don't know, you guys know Paola, always laughs at me because I just get very frustrated, and I'll just say, like, these are just words. And the reason that I focus on words is that until we can kind of agree on what words are meaning, we're just talking about nothing. We're just saying things. And so you'll see people argue about this, like, what's love? Like, it doesn't, you can't have a conversation about it if you're both just talking about different abstract concepts. And so the more we can be precise with our words, I think the better we can communicate and the more we can do good science. And George Orwell has sort of a great quote that I quote over and over and over. It's that, if thought can corrupt language, so too can language corrupt thought. And I think that's very important, right? Because when we start using words like depression, we're already sort of tinting our thought process in it. And so when we're talking about, life, what is life? Maybe the word doesn't matter and we should just sort of go after what we're really talking about. What is the actual objective value of the things that we're talking about? All right, so there's 4 levels of organization, right? The primary structure is just, the linear chain, right? We've got our linear chain here. If I were to just go through and name the side groups, bam, 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 it's sort of long and whipping around. That's our primary structure. Bailey, glutamine, whatever. The secondary structure is formed by hydrogen bonds between the backbones of these things. Okay, so you can see here, here's the beta-pleated sheet, here's an alpha helix, okay? So initially it's a long chain as it's being made and it's just whipping around. Temporary structures get formed by stabilizing bonds. So this beta-pleated sheet is now kind of semi-stable as it's whipping around, and this alpha helix is kind of semi-stable as it's whipping around. In the tertiary structure, and again, we talk like it's linear, this is all happening at once, but it's just easier to think about it this way. The side chains can begin interacting. Polar kind of wants to be on the outside, non-polar kind of wants to be on the inside, and they have attractions or repulsive forces with each other, and so you get this eventually stabilizing into a relative shape in a certain environment. So that's what our tertiary structure is, and that's what it comes from. These things are largely dictated, again, by the backbone and this ultimate folding. If we had to guess, what's this one? Is this polar or non-polar? Polar, probably, right? What about this one deep in here? No, right, so again, just rough guessing, that's probably what's going on here. And then you can have one protein going with other proteins to make a quaternary structure, right? So most of the proteins, well, a lot of the proteins, rather, of interest to us are not just made from one protein. Like GABA has 5 receptors, right? You have 2 alphas, not 5 receptors, 5 proteins that make up the receptor. So you have 2 alphas, 2 betas, and a gamma, generally. And then there's genes for 5 different alphas, and probably more than 5, maybe 6 different alphas, 4 different betas, different gammas. So you can have 1 alpha 1, 1 alpha 3, so there's a lot of different variable expression for our ultimate protein desired product because of that. All right, so let's get ahead of ourselves a little bit then. How do you generally activate an enzyme? What do we think about happening chemically when we activate an enzyme? There's a few ways to do it.
Phosphorylation.
Phosphorylation, that's absolutely right. Phosphorylation, dephosphorylation. So what is a phosphate group?
PO4.
Yeah, right, PO4. And there's a lot of what to that?
Negative, I guess, negative electrons.
That's right. So it's going to be polar or non-polar.
Polar.
Polar, right, largely polar. So let's say I come in here, and what I'm getting at here is that this is not a linear additivity, and we'll see more of this later. If I come and I put a phosphate right here, okay, what's going to happen?
It's going to change the structure.
Like entirely, right? I'm just going to rip this off to the side, and who knows how that changes this shifting and this shifting. So one small change in the right place can create a huge conformational change in these things. And that's called, and it can be called different things, but allosteric modulation usually is when a small molecule can go in and make a big difference in certain protein to affect its function. Okay, good, so we can start thinking, right, what's going to happen, those sorts of things. And then again, just this idea, within the environment, I change the environment a little bit, you whip these things around and everything can sort of change. All right, so we'll just see a quick video of that. I'm actually doing okay on time.
Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to coil and fold into unique three-dimensional formations. Two kinds of secondary structure units are the alpha helix and the beta-pleated sheet. The folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as the quaternary structure.
Okay. So it's hard to find a really great video for that, but that kind of gets the job done. All right, so now I'll pretend like I'm going to make this clinical, and we'll talk about sickle cell anemia. Okay, so here's a normal hemoglobin protein. And as we can see, it's got a quaternary structure, right? So it's 140 something 6, maybe. 141 amino acids long, right? And you know, we could just write out the whole long sequence of it, and then it goes on to make the secondary, tertiary, and then 2 beta units and 2 alpha subunits ultimately makes our hemoglobin. What does hemoglobin do? A lot of things, but carries oxygen, right? Okay. And it's sort of a protein within a red blood cell. So it flips around and it carries our oxygen and it does all those things that we kind of want oxygen to do. Okay, and so everybody's happy. We have this sort of healthy, nice red blood cell and everything's great. All right, what happens in sickle cell anemia?
It's a glutamate for valine substitution.
Yeah, it's a glutamate for, or glutamate for valine substitution at amino acid 6, okay? So it changes, that is a change in the what structure? When we say that. I'm sorry?
For the primary?
Primary structure, right? And then that obviously will have repercussions down the line. So we can see it is significantly changing the shape of this, which is then sort of changing the quaternary structure of this molecule. Okay, so I want to point out a few things. There's 141 amino acids. One change, okay, creates a huge difference in the structure, and that's something that we see a lot, and that helps us in evolution. We can do little tweaks and get big changes, which lets us play kind of with function a lot, which is good, but sometimes it leads to things being sort of not necessarily what we desire, but we'll talk about that in a little bit. Secondly, how could this happen? If we were to go before the protein was made, how come that happens?
Something like genetically or how?
Yeah, sure.
Like when they're sequencing it, like when it's going through the ribosomes and sequencing everything.
The reading frame, right? We're flipping through,
Exactly.
Yeah.
It gets to 6 and what happens?
It's like a missense mutation.
I have
Is
to
it
go back and read
sense
it.
or
Nonsense or
nonsense? One of
missense?
those.
It's not a nonsense. Oh, is it a missense?
Yeah.
I'd have to go back and look. It's a single amino acid change.
Change,
Is that
yeah.
It would
missense?
have to
Yeah.
make sense.
I'm embarrassed because I don't know. Yeah, we'll call it a missense. Anyways, it's not a nonsense for sure, and it's not a determination, so it probably is a missense. I should just trust the med students. And so likely one of those amino acids, and I don't remember exactly, one of them is, I think it's an A to a T or a T to an A, and that just changes the reading frame, not changes the reading frame, it changes what amino acid sticks, and it's just a different amino acid. So that's originally where it came from, and then we get this big different structure. And not only just quaternary change, but sort of the function change, and that's what's more important. These usually flip around just fine. These sort of clump into these rods or fibers, which changes the macro shape of the cell, which then changes its ability. Usually our vessels can get very nice sort of single vessels through it. This can clump and cause kind of ischemia. It doesn't effectively transport hemoglobin, and that can create the problems of sickle cell anemia. Okay, so that's sort of, oh, and the other thing is that do people with sickle cell always have sort of problems? What triggers that?
Stress, different
Infection.
dehydration.
The environment, right? So already seeing like environment is very important to this sort of function. What's going on within the cell is very important for this sort of function. So, all right, just sort of pointing out how one single amino acid change can have a huge impact on every single level. Protein, primary expression, tertiary expression, function within a cell, and then life of an individual, right? How many bases do we have in our DNA? I don't even remember, but it's like 3 billion, I think, 2 billion. It's something, it's a lot of billions, a couple billions. I think it's 3 billion. So one base pair change in 3 billion can create a very different life. And so cystic fibrosis, right? One base pair change at 3 billion. You start mucking up the Krebs cycle and really important stuff, it's not compatible with life. Some bases you can just go nuts and flip and change, and it's not a big deal. And you know, it matters where things happen, I guess is what I'm getting at. So this is not psychiatry, okay? This is a much more simpler process to understand. But psychiatry works on the same rules. So I want to talk about a couple of things. One is that this is only a disease because we call it a disease, okay? It is just a mutation. And why, so sickle cell is not a bad thing in the evolutionary sense, and I can say that confidently for a few reasons. Do you guys know why?
The blood cells have a shorter lifespan, so they're less likely to get malaria, die from malaria.
Right, so the protection from malaria. But how come I don't think that that's just a random thing? I mean, we could just have that randomly happen, luckily.
Because it continues to survive.
It continues to survive, but why else? There's one big reason. If you see this happen in evolution, you know that it was a pressure for it. You know that it's pushing for it.
It's in a specific population, and like the heterozygotes get the most benefit from it.
Again, this is one of my thinking kind of questions. It happened a lot. This mutation happened multiple times over time in different regions independently, and it happened in different ways. It's not only this mutation that can do this. Thalassemias, all those. We had lots of push for this. So what does evolution care about?
Survival.
Yes, but for what? Why survival? Not just survival. It doesn't really care about survival, except for what? For reproduction.
Yeah.
It's a numbers game. That's what it is. It's a numbers game. And so sickle cell anemia is not a disease. This evolution, and it is a disease, and we'll talk about the semantics of that a little bit later. I'm parsing my words now. But what sickle cell anemia, what this mutation is a cure for malaria or resistance to malaria. Okay, what came along with it was the potential for this. Okay, so we have to start understanding that is how genes work. Genes aren't simple. Sometimes to have a system, you got to bring things along with it. All it cares about is numbers and risk versus reward. Okay, that's what's going to get passed on. So let me just draw a couple of things really quickly to kind of point that out. And this is simple because it's sort of a Punnett square. Okay, we're going to draw two different situations. All right, and whatever. There's going to be 100 babies in each of these. And I don't even remember. Let's just say this is I should probably know what this is. We'll just do, and S is not good either.
HBSC and HBS.
But I can't make capital versus not.
Oh, yeah.
Well, we'll just pretend that, I'm sorry. And this is going to be all capital. Okay, so this is the population of 2 healthy people mating, and then 2 people with the sickle cell trait mating, okay? And so 400 of each get born. And let's say 50% of the people get malaria and die from it, all right? So in this situation, this is going to be capital S, capital S. This is capital S, lowercase s, capital S, lowercase s, lowercase s, lowercase s. Okay, these people here, 50% get it and die from it. How many live?
50.
All right, we're going to draw the sort of outcome from this. All right, I should have done this beforehand so I don't mess up. All right, this has the trait, and let's see with the trait, 10% get it and die. How many do we have?
90.
90. All right, and this one here, the same thing, right? And then whatever, we can make up a number. Let's just say 90, and we'll put a 90 star, right? And the star means that they also have sickle cell. This one here all has capital S, capital S. So what does this look like? 50-50-50-50, okay? So we can immediately see a couple of things. In this population, there's going to be 200 people who survive, and there's no sickle cell, okay? In this population, there are 320 people that survive unless sickle cell kills someone, and 90 of them have sickle cell, okay? Even if no one with sickle cell, even if it was a mutation that stopped you from being fertile if you had this, evolution would still push. I mean, again, it could be more complicated than this, but just to generally see, evolution doesn't care about your suffering, right? It cares about these numbers. That's what happens. If the numbers push, it sticks. And the reason that this sort of works is because we see it arising multiple times. So we know that likely it provided an evolutionary benefit because it arose multiple times and it stuck around. So the reason I bring that up is that evolution in psychiatry gets really complicated, and I hedge away from evolutionary psychiatry, but I think it's useful if people aren't being ridiculous, and people are sometimes really ridiculous with evolutionary psychiatry. So I'll give my basic thoughts on it. I think that if you want to create a system that has a one-to-one response character, a rigid system, you can do it without breaking. If you want to create a system that has no flexibility, that's rigid, you can do it. If you want to create us and be flexible, the price you pay for that is that the system can break. There's just no other way around it. With flexibility, you buy the possibility to break. And I think that's just a necessary part of the system. I do not think that biology is able to create a system that is flawless and also flexible. I just don't think that that's a possibility. So when we see depression, okay, I think depression is just going to be part of us. And that could be for two reasons, right? It could be that depression is the sickle cell anemia price that we pay for the flexibility that lets our numbers get passed on, which is probably the larger part of it. And the second could be that depression actually offers an evolutionary advantage. And that's something that can get you into trouble for saying, but I think that there actually is some validity to that because of animal models. And the other thing that you have to realize is that what evolution is looking at is populations. It's not looking at individuals. So if you share 90% of DNA with something else, and that other thing, your disease makes that other thing survive more, it can be beneficial. And that gets into complicated things, and you look at like social defeat models and what's the benefit. So it gets complicated. I'm not going to talk a lot about that, but just this idea of our system being flexible and as a necessity of that, having this ability to break. It's kind of like a no free lunch sort of thing, and I think that that's absolutely true of the brain. So I think that, that being said, at this point in our civilization, depression's an illness, okay? Even if it was evolutionarily advantageous in the past. I'm not going to call it a disease, but I'll say that it's an illness, it's suffering, and our goal is to ameliorate suffering, right? That's the goal of the doctor. So we definitely want to kind of treat it, but I think that it's understood really probably as a different way. You're not going to find a single gene for depression. And the reason is that depression is a possible outcome of a complex system. And I think that that's true for anything that you see in large amounts of the population. Anxiety, right? Anxiety has its place. Pain has its place. Even schizophrenia has a far out on a spectrum of possibility of creativity and perception is necessary. And we see that schizotypal magical thinking has its place. It has a beneficial effect in groups. And so kind of getting us to think a little bit differently about these things, I think can be useful. And again, getting at this idea that evolution is a cruel mistress, right? We care about suffering, but evolution doesn't. Evolution does not care about suffering unless it directly has a negative impact on your ability to propagate. And I think we have to keep that in mind when we sort of are looking at these things. Okay, so that sums up proteins. I've come through one of the four, and I've got like 20 minutes left. So anyways, proteins are the workhorses of the cell, pretty much. They get stuff done. I'm going to fly through this stuff really quickly. Carbohydrates, okay, mainly used for energy storage. In plants, starch. Cellulose is mainly a texture or strength in plants. Glycogen is a branch chain carbohydrate in us where we store our energy. Just kind of looking at, monosaccharides, disaccharides, okay. Where else do we see sugars besides in energy storage in us? We've seen a couple of things. I just talked about evolution, which is about what?
Reproduction.
That's not reproduction, but what is the initial or the main component that gets passed on, or the main building block of
DNA.
DNA? All right, and what is DNA made of?
Sugar.
Ribose sugars. Okay, so what's a ribose?
That.
It's a sugar, right? We said that. How many carbons does it have? All right. This is a ribose, okay? Actually, that's ribose with a, anyways. So RNA is ribonucleic acid. All right, here's the base, the nucleic acid base. Okay, here's the ribo. DNA is what?
Deoxy.
Deoxy. So what do? You get rid of an oxygen, okay? That's RNA to DNA. So we can start thinking about these things, and it's got, whatever the, how do I even draw this? Actually, that's not there. It's like that, I guess, the phosphate. Something like that. I don't know. I don't know. Close enough. That's the point of it. So it's also incorporated in the DNA chain. We'll just keep moving on. All right, what are these?
Fatty acids.
Fatty acids, right? Are they polar or non-polar?
Polar.
How come you say polar?
Because they have the carboxyl.
They do, but you're right. So were this not here, would it be more or less polar?
I mean, non-polar.
All right, so it's a relative spectrum, right? On the big side of things, we would call it non-polar. But you're right, this makes it more polar than this would be, but relatively, what we're sort of seeing is, and when it reacts, what part do you think reacts? That part, absolutely, because in our next slide here, right, we've got glycerol, which is going to form our triglycerides. Look at what part's reacting, and that's, you know, we can predict that, right, from sort of looking at it. Again, it's a hydrolysis reaction, we do water chemistry, we get triglycerides. And then, you know, if you look in the cell, this is what you sort of see. So when we check triglycerides, this is what we're checking. Okay, so as a clinician, how much do you need to know this stuff? Probably not much. Probably not at all, really, if you're just a strict clinician. But at some point, for you to be able to order triglycerides, somebody had to understand the structure and understand how to assay it. So if you want to push the field ahead, you have to know stuff. If you just want to be a clinician, which is fine, and it's fine to just be a clinician, there are other things that you need to know. But I think it's useful to know this stuff, or at least cool to know this stuff too. And it also kind of gets at etiology, right? When we see somebody die, we do an autopsy, how do we gain knowledge? We just sort of muck with stuff, we see differences in statistics, we see, hey, what's different here? We scoop this stuff out as triglycerides, we start checking it in blood. It provides a putative mechanism to treat, right? Can we treat these things? Do they help? Sometimes we trick ourselves and we think, oh look, triglycerides are high, I had a heart attack, and we treat triglycerides and it doesn't make them better. But anyways, that's how we sort of move science ahead, and medicine ahead, is by knowing this stuff. So what do we care about phospholipids, or lipids rather, because they can form phospholipids? Again, glycerol, 2 fatty acids, and phosphate group. Does anybody see what's wrong with this in one of the nonpolar tails? Why is this, somebody made a mistake here? I didn't see it until today. I've looked at the slide many times. What's the valence of carbon?
Oh, 4.
4, right?
Yeah.
Oops, we broke a valence shell there. But anyways, and you can see here that we can see why this arranges. And if you actually sat and thought about this for a long time, and I said, hey, I'm going to take this and I'm going to squirt it into water, and I just want you guys to think about what's going to happen. What's going to happen? What are they going to want to do? You could imagine that they would form a double membrane. Right? That is the conformation that it is happiest with. And if you sit and think about it, you would start thinking, okay, polar must be non-polar, or does it just form a line, and you would want to minimize surface area or whatever, and this is what you would come up with. So that's how we form sort of our membranes. Okay. Anybody who knows me has probably seen this picture. I like it a lot. It's very useful. There's colors, etc. Cholesterol is not a bad thing, but it is a lipid. And again, we can see here, these are enzymes, which are same thing we're going over and over. Enzymes are proteins are made from whatever. And if you want to make a certain thing, you just express these. And the ones that you might hear about is 3-beta HSD, or hydroxysterol dehydrogenase, 3-alpha HSD, which actually shows up here, and 11-beta hydroxylase is probably involved in the metabolic syndrome with atypical antipsychotics. Just sort of things that might pop up in your career in psychiatry if you go that way. But again, it's just a great slide, great learning tool. Cholesterol is a lipid. Okay, nucleic acids, sort of looking at this. What are each of these things here? Each one of these circles, what do they represent?
Hydrogen?
Not necessarily hydrogen, but what is hydrogen?
An atom.
An atom. So each of these represent an atom. This one here is, you don't know, but you can maybe guess. It's a hydrogen, and that's asking too much for you to do this. It was adenine and guanine, thymine and cytosine, the 4 bases. These are purines, they're different. They're sort of 2 ring-like structures, and these are each 1 ring-like structure. These are the phosphate groups on the back. These are forming hydrogen bonds. That's all that really means. And so this is what the nucleotides actually look like. And then you can kind of see polar on the outside, non-polar on the inside. So when this thing kind of organizes, you've got kind of the greasy middle. It's kind of like oily, so it wants to stay away, and then the phosphate's on the outside. This is a very cool structure. DNA is a super, super cool structure. And the way that Watson and Crick kind of ended up describing it, also very cool, just the science behind it. So we're going to do a quick discussion, and I actually think I can get through what I want to get through in the next 15 minutes. So, favorite protein? Anybody have favorites? I pick favorites. No? We can talk about it or think about it or sort of... Okay. I like actin. It's probably my favorite protein. I I
do
did
like
ATP
actin?
Why do you
synthase.
like actin?
Actin is a fascinating molecule. It really is. It can self-assemble in very complicated ways. It's used for lots of different things. Actin is really probably at the core of consciousness in some ways, obviously with a lot of other things, but actin's a very cool dynamic molecule. And I'm into dynamic sorts of things, stuff that kind of has a lot of flow to it. ATP synthase is very cool because it's very like, it's like the smallest motor you can imagine, and it really is a motor. It's like a rotary motor. But also, I like receptors. Receptors are pretty cool. Fatty acids, we talked about the omega-3 fatty acids, omega-6s, omega-9s, I went over those. You can look at the chemistry of why they kind of do what they do and why there might be a connection with inflammatory disorders or psychiatry or heart disease or whatever by how they get into the icosahexaene pathway with enzymes. I'm not going to go into that a lot. Anybody have any favorite evolutionary psychiatry theories or thoughts on it? I try and really hedge on evolutionary psychiatry. There was a resident who graduated who would say these just things, and they just didn't make any sense. So really talk about evolutionary psychiatry, you better know genetics, at least a lot. Because if you don't, you're just making stuff up. And you need to at least understand how the genetics game works and the evolution game works. And it's complicated, really complicated, particularly for psychiatry. But you hear lots of stuff about evolutionary psychiatry, and it pops up. And it's dangerous because people can start being really judgmental about things. And then emergence. Do you guys know what emergence is?
No.
Okay. So it's getting it's something arising that did not exist from the individual parts. It's the idea of the whole is greater than the sum of its parts. And that's really what we're getting at. We're building towards a brain, right? And what we're trying to get at is emergence. How do you take these proteins and then make a behavior from it? And we can get pretty close to doing that from a behavior standpoint. Consciousness is a lot more complicated, but we can begin to think of it, right? We can begin to think about moods and emotions and behaviors, really using emergent property of neural circuits. And I think that's really where the field is heading, and that's where it needs to head, because talking about neurotransmitters is really, really just taking a snapshot that's very wrong, I think, in these pictures. And the idea of even just brain modules only recently being kind of incorporated, and incorporated in the wrong ways as like static models of doing things, is just very wrong. Emergence is really, how do we mix colors and flavors to kind of get what we need to get? So okay, I'm just going to introduce thermodynamics. That's really all I wanted to do. And then next time we'll sort of finish thermodynamics. So, all right, these guys all love thermodynamics. Do you know who any of them are? This is Prodigy. Song? Firestarter. This is Jimi Hendrix. Song? Probably a few. Let me stand next to your fire. This is like the most awesome, although I didn't know their names. It's Rockmaster Scott and the Dynamic 3. You know the song? The roof is on fire. Look how awesome these guys are. These guys here, AC/DC. The house is on fire. Her, Adele, the song. Set fire to the rain. This one here.
Alicia Keys.
Alicia Keys. She wrote this song about my, it's kind of funny, true story. She wrote this song about my girlfriend. What? Name of the song?
Girl on Fire.
Girl on Fire, right? Jennifer Lawrence. But I'm lying to you. She actually didn't write it about She didn't write it about my girlfriend, Jennifer Lawrence. She wrote it just kind of as inspiration, as like a confident woman. All right, so we're just going to run through this really quick. Getting at this idea of conservation of energy, the first law of thermodynamics. Is anybody kind of, what is conservation of energy? Just in a nutshell.
It's not created or destroyed.
But it can be. That's right. That's energy cannot be created or destroyed is the first law of thermodynamics.
Yeah.
But implied behind that is that it can be transformed. It can be moved around. It can be shuffled. It can be manipulated. Entropy. Who knows what entropy is?
Randomness.
Randomness. Largely, it's the amount of freedom that a system has, which is usually determined by random interactions. It looks like randomness. It's really disorder in a system. Okay. So again, where did this start? Which is the starting state between the two of these? If these are gas molecules,
The right.
the right is the starting state. That's absolutely right. And sort of getting at the same idea, if you toss bricks off the truck, which pile of bricks are you going to end up with? The right, okay. So entropy is where things push. And I'm going to get a little bit more detailed than that. So again, This is an increase in entropy. There's more heat in this system. It has more freedom. Okay, there's more movement. All right, here's just another sort of example of entropy, right? So what is this, if I allowed this system to continue, what would we ultimately end up with? Likely, at least.
Water.
I'm sorry?
Water. Do you mean which way?
Let's just say these are gas molecules. If I let this system continue, what would it look like?
It'd even out.
Even
It would even out. Okay, good. So this is probably the most important slide on this whole talk, okay? If I can get through this, I don't know whether to start the next section or not, but this is, if we can understand this, I think it's an hour worth your time, okay? So this is getting at diffusion, all right? What force drives diffusion?
Concentration gradients?
Okay. That's a good answer. What force drives it? If you want to manipulate, if you want to predict, if you want to be a scientist, you do want to know concentration gradients. But what's the force that drives it?
Disorder, entropy, I don't know if there's.
That's a good guess, because that's what we were just talking
It's
about.
like-
And you're right. But it's not a force. The answer is that there's not a force that drives it. Okay, it's just the way things are. Entropy is the phenomenon that we're kind of describing. So let's just pretend that this is a dye molecule in water. Okay, why is it bouncing around?
Hitting the water molecule.
It's hitting water molecules. It's Brownian motion. Okay, it is largely a random phenomenon. Right? We can consider it a random phenomenon here. This marker here is a barrier that is impermeable to our water and our, or it doesn't matter water or not, to our dye molecule. Okay, so I want you to just watch this dye molecule until you can convince yourself that it is moving randomly. Okay, there's no force driving it. At any moment in time, it is just as likely to be moving up as down, as left, as right. And it is simply the combination of Brownian motion and millions of interactions that's dictating it. The previous state, and what I'm saying is the previous state is not determinant of the future state. At every moment, its interactions are going to be, for all intents and purposes, random. Are we convinced of that? Okay. So, is there any force driving that? Okay, is it acting randomly?
Yes.
Okay. We're going to come down to here. There's 100, I don't know how many there are. And just watch one, okay? It's hard to watch one because you can't necessarily follow it. But I want you to kind of be convinced that they're moving randomly. Describe to me what's happening as this is being removed.
Well, they diffuse apart.
They just occupy the whole
Yeah.
space.
Okay, they are.
diffuse into it.
So you're right. They're now occupying that new space. Anything else that you're noticing? So the relative concentration of this new space before the potential for something to be in here, the probability for something to be in here was what? When there's a barrier. What's the probability for something to be on this side while the barrier's up?
Nothing.
0.
It's 0. When this is gone, you've created a new potential space. And so randomness now has the potential to infiltrate a new space. And at that cost, you have a new distribution of particles over a larger space. And then you're seeing a relative what in concentration of any individual sort of place? Decrease, okay. Now I need you to convince yourself down here that there are a billion molecules, okay? And that what is happening here is random. Every single particle in here, every single billion of them is moving randomly, okay? It's moving randomly. There is no force that is driving this. It's random interactions that's driving this. So this is a really, really simple concept that is amazingly difficult to grasp. Is that believable to you all? Okay, so a few questions, okay? If we have one particle up here, and I were to draw a line in the middle of it, and say, what is the chance that every particle will be on the left side after this is run for a minute? What's the chance that it's going to be on the left side?
100%.
Is it 100%?
With the line?
No.
No,
Let's
without
say
the line.
let's say it's
without
all
the
open.
line.
I drew a line that it could cross, but just an artificial line.
Oh,
And I said,
percent.
if I pause it after a minute,
Oh, okay.
what's the chance that it's going to be on the left side?
50%.
Okay, let's say there are two particles. What is the chance that both particles are going to be on the left side?
25%.
25%. Okay. What if there were 20 particles?
20 to the whatever power.
Okay. What if there were a billion particles?
20 to the whatever power
Okay.
again.
Right, not exactly that number, but yes, we're starting to look at astronomical things, okay? What I want you to understand is that if I let this run long enough, over an infinite time period, you would see this, if you let it run long enough, could potentially be just in this corner. And it would, if you let it run long enough, because there's not a force. It is a statistical distribution, and this statistical distribution is not fixed. If you let it go, and the numbers are so high that given the time of the universe, you're not going to likely see them. The reason I say that is that actually, these types of interactions occur in our body and are important. Our body works on statistical mechanics. This is a statistical mechanic. We can predict what's going to happen here, right, with a level of certainty. We're getting at levels of certainty. It's a matter of how certain, okay, and that's kind of what science is. How certain are we that things are going to happen? This is how, again, these are random. You will get this because that's how randomness sums to create a predictive pattern. The reason I bring this up is that that's how our brain works. We have hundreds of trillions of synapses, and they are not acting orderly. They are able to sum, right? If you look at this one thing, if we had one neuron, we'd be totally random. The reason we can have orderly behavior is that we have enough that by chance, given a situation, we have the ability to act in an organized way. This is how neurons work. If you watch an individual neuron, an individual synapse, it is chaotic. It is this. It is only when you have billions or trillions can you solve problems. That's why I'm spending time on this. Because if you're looking at a neuron, you're missing the picture. You have to understand what is the drivers. And so this, if you let it go, you could predict pretty comfortably what will ultimately happen after an hour of this. If we just let this run for an hour, what would ultimately happen?
It'll all be the same color.
It'll all be the same
Yeah,
color.
completely.
That's because there's no force driving this, okay? So it will go to the highest level of entropy, which is a full distribution sort of distance between particles. Our brain does not have that. Our brain is constantly being flipped from order to disorder, and that's what we do. We thrive on that border of order and disorder. Okay. One last question, right? If you are looking at just this one particle, or a bunch of particles, moving randomly, if I were to zoom out further and further and further and fast enough, what would it look like?
Just a point.
Just a point. Okay, I'm glad. Can you really believably see that? Because if you can, you understand what the process, because you're right. It depends on the speed at which it's moving and the speed at which we're zooming out. And you can actually you can use mathematics to calculate what is the probability of the distance that these things will move. So it's very, to me, these things are very cool. If you can actually see that through random movement, if you can zoom out far enough, you will see, because that's what we're kind of getting at, right? What are the levels that we're kind of seeing? If you can actually zoom in far enough in here and see this, because that's the exact same thing that we're talking about, right? It's the flip on its head of that. If you can see that, you got it, okay? If you can get it, I think that it is a valuable lesson, because this is how the body works. It's very much not what we're taught. We're taught linear reductionism. This is really how nature works. Okay, so that got me close enough to where I wanted to get. Do you guys have any questions before we wrap up? Okay. Well, thank you for your time, guys.
Thank you.
See you.
Thanks.